Hello, everyone. Welcome to the CannaBuzz podcast. We are broadcasting live to the CannaBuzz community. Shout out to everyone in the CannaBuzz community that's watching us live right now. Without you, we wouldn't be possible. And if you're watching us on YouTube, thanks so much for watching us on YouTube. Make sure to like and subscribe so that you can keep up with more videos like this. Today we have a great one for you today. We have our friend Jackson, Farmer Freeman. We're going to be talking all about Hop Latent Viroid testing and also what Hop Latent Viroid is and all kinds of deep dives into viroids. So stay tuned for that. He's got a lot of great information for you and a lot of great questions put together by JR. But I need to get through a couple quick shout outs. So let's do them real quick. First off, shout out to our friend TikiMadMan. Tiki just sent me some seeds, and we're going to be giving away seeds to our community members. And also, we're going to be giving away a seed pack at the end of this show from one of our friends. So, to our live viewers, that's another perk for our community members. So, anyways, shout out to TikiMadMan over at TikiMadMan.com and Tiki Seeds. And JR, what's the latest from from Tiki?
Well, one of them that's doing really well right now is the abdominal runts. And that one's available at tikiseeds.com. It's also available at neptuneseedbank.com. And the abdominal runts is a Y-Life by White Runts. So it should be a frosty girl. And like I said, you can pick those up at tikiseeds.com or neptuneseedbank.com.
Awesome. So shout out to TikiMadMan. We really appreciate you. And like I said, he just hooked me up with some seeds, so we will be giving away those soon. And we also have an interview with Tiki coming up in about a month. So I'm probably going to hold on to some of those for that one. So also shout out to Terploc, our friends over at grovebags.com. If you want to cure your weed, check out these bags. They make them in all sorts of different sizes. This is their glass jar size. So you can basically replace one of those glass jars. I can replace I've got a whole storage container of glass jars that I need to figure out how to get rid of because I've got all these Grove bags. They make them in all sorts of sizes, and what they do is they cure your weed to perfection. So check it out. It's grovebags.com. Use the code CannaBuzz for 7% off your order. And thanks to our friends over at Terploc and Grove Bags. And lastly, shout out again to the CannaBuzz community. Use the code GrowersLove for 50% off if you want to join our community and help support us and make more shows possible. All right, Jackson, thanks for hanging through the shout outs to all the homies that make this place possible. But I'm super stoked to have Jackson on the show. We did an episode this past fall, I think it was, where we talked about sex testing, and then we've interviewed you even I think a year or two before that one. So we've been keeping in touch. Always learn something new every single time. But let's get into your official introduction. We're going to dive a deep dive into the world of viruses and viroids. Our guest, like I said, is no stranger to the show. He's been on many times before. So let's dive deep into the world of viruses and viroids. Welcome to the show, Jackson Farmer Freeman. Oh, you're on mute, man.
I am on mute.
There we
Thank you,
go.
Q, for that introduction.
Yeah, no.
Yeah, glad to be back on the show, man. Yeah, since we talked last time, we've made a little bit more headway into this world of viruses and viroids and getting the tests out there for everybody, so that they too can learn about how it's currently affecting their gardens, or hopefully not affecting their gardens. But yeah, thanks again for having me.
I think the timing of this is going to be great because a lot of people are getting those cuts and getting everything ready to rock for the summer season. And now they could just have a little ease of mind, dip it off, get a little testing done, make sure they're good to go, and hit the ground running. So
Yeah.
timing, I think for this is really good timing. And we're super excited to have you on. I know you have a little bit of a 420 sale kind of lingering on till Monday. And so could you talk a little bit about the sale and your lab services and what you offer there at the Farmer Freeman company?
Yeah, sure. Thanks. So, We're a molecular biology and genetics laboratory. We're based in Washington, D.C., on the east coast of the USA. We've founded in 2015 and been offering our first product, the EasyXY plant sex test, since about 2017, after a couple of years of testing and validation. So yeah, in addition to that sex test, which is like our flagship product, we've sold so many of those, saved so much water and soil for growers around the world. In addition to that test, we're now offering viroid and virus detection tests. And then in addition to, botrytis, fusarium, powdery mildew, just your most common pathogens that are going to be found in your cannabis or hemp garden. We have detection tests for those. Um, in addition to the things we do with cannabis and hemp, we've done a little bit of research and now offer sex tests for different like specialty fruit trees, including kiwi and fig. So
Cool.
these can help you save years of waiting to know the sex of your fig tree. Um, yeah, it's just super important for if you're making a fig cross, things like that. There's actually a huge community of fig growers and
Interesting.
enthusiasts out there. Yeah,
Yeah,
there's
it's awesome
Uh,
that things are getting that way to where we have all these little niche communities that we
yeah.
can all learn from and kind of get community with. It's really cool.
On the sex
it's
testing,
awesome. Yeah.
on the sex testing bit too. So I almost forgot to mention at the top of the show, it was just 420 the other day. So shout outs to everyone. Hope you had a great 420.
Yeah.
But the other day I was talking to some farmer friends of mine that were at a 420 event. And these guys, grow a bunch of plants in Northern California, and they don't sex test their seeds. And so I was talking to them. I just popped some of their seeds that they sell, and I was like, "Yeah, I'm gonna sex test them like in a week or two." And they were asking me about it, and so their questions were, A was, do you sell sex tests in bulk? And then maybe could you speak to just like a use case where like a farmer uses your service, your sex testing to help them out?
Yeah. So yeah, we've heard that before too, where people are still, even though we've been out for over 6 years now, some people are still just learning about the DNA
Yeah,
sex test. Um, or maybe they heard about it, but they weren't understanding how it's different than just, taking a clone and putting it under 1212 and finding out a couple weeks later. So yeah, the DNA sex test, you can tell the sex of your plant when it's just about a week old. We only need like, a fingernail size piece of leaf, or even those first round leaves, the cotyledon, those work great. From that leaf, we'll take the DNA out and do a PCR test on that DNA sample, looking for male associated DNA markers. Um, so yeah, we've heard, outdoor growers especially not using the test. And I think, in some cases it makes sense. It's not a free test,
right.
you are spending typically between $8 and $12 per sample, depending on how many you're doing.
Okay.
Um, so farmers who are growing hemp from seed, it may not be worth it, especially if they're not sure even what they're going to do with the hemp. If they're just going to grow a bunch of biomass and extract it, it
Right.
may not be cost effective to
But
get rid of the males.
if you're growing a
Um,
bunch of great flowers, though, you don't want to be. And then when I was talking to this guy, Johnny, it was about how it could save him time. And he was saying this. He's like, oh, that's like a couple times that I've up potted it. I could, replace by basically sex testing it.
yeah. If you're using, some living soil that you've had for decades, it's valuable. You don't want to waste it on plants that you're not going to keep. Then definitely, consider testing before you up pot and spend another, 3 weeks worth of water and all that real estate that those large pots can take up.
Yeah.
I
So
have
yeah, thanks for the
I was,
detour there. Sorry, JR.
yeah, it's okay. I was wondering about the thing that rang out in my ear is the $8 one. And how many do you have to buy to get it for $8 a test? Just
So
curious.
that's 1,000 or more samples.
Okay. Okay.
And generally when you're doing over 1,000, over 2,000, you're not just going on the website and clicking buy now. We'll talk a little bit first, figure out exactly what your needs are, and develop the price point at that time. But that being said, we do have some customers who will go to the website and buy the 1,000 pack, and we love them for that.
Yeah, yeah.
It's a
Love
good
them for that.
surprise.
Yeah. And then, in theory, if you wanted to do like 100,000 plants, it would again be significantly cheaper than the $8 price point.
All right.
So yeah, we can definitely we can handle bulk orders. We can test about 1,500 plants a day comfortably in one 8-hour shift. We can always work overtime and get those large orders done
Yeah.
even faster.
And
Uh,
you guys are super quick. I mailed some samples in back in like January or whatever it was, and I got results back within a day or two of you guys receiving them. So super fast. But JR, let's get back into the. Sorry for the detour on the sex testing. I just wanted to mention it because it came up
no,
the other
sure.
day.
I mean, yeah, no, it's good to know that these tests are available for just regular home grower consumers and commercial users. We also have our sex test and our viroid test in retail package versions like you see behind me, that you can buy at your local grow shops as well.
Red and
And the
ask around.
420 sale is going
Yeah,
until
yeah.
Monday. So
Yeah,
don't miss out
thanks.
on that. That's some really great deals. I got to order some testing myself for some of my moms. I want to make sure my garden is super clean. But yeah,
Yeah,
so we'll go ahead and move on. And so as we're on the subject of viroids and viruses, can you kind of explain a little bit about the differences between the two?
sure. Yeah. So, I mean, I think most of us are familiar with viruses since, we were young. We've heard about the flu virus, when I was a kid, the swine flu virus was a thing. And so that, present chicken pox, obviously. So we've been presented with a bunch of different viruses throughout our lives. And now when you're in the plant world, you learn that there's other things called viroids. And so these are similar to viruses in that they're an RNA-based structure instead of a DNA-based organism. And viroids only infect plants. And the main technical difference is that they lack a protein coating around that RNA sequence. So that makes them a lot smaller. And because they were smaller, they also were detected and identified, after viruses were. They were a bit more mysterious for early scientists. So yeah, because they're small, they lack that protein coat. It's definitely one of the most, primitive kind of life forms on the planet.
Interesting. So, can you talk us through the techniques that you use to detect viroids or viruses in a sample? I know like when we were talking about with sex testing, I can just clip off a cotyledon and throw it in an envelope and call it a day. But how does it work with detecting viruses, and especially on your end? Yeah, what are those techniques to detect them?
Sure, yeah. So on your end, the consumer end, the sampling protocol is going to be really similar. You're going to send us a couple of different leaves from different areas of the plant. In addition to, we'll often request root samples for certain viruses or viroids. And the reason we need multiple samples instead of just that one tiny piece of leaf is that we're detecting the RNA from the viroid itself, which may not be found in every single plant cell or every single area of the plant. That DNA sequence for the plant sex, I mean, that's going to be in every single cell of the plant. They all have that same blueprint that tells, the organism what it is. But the RNA and the viroid can be kind of localized in areas of the plant. And that's also why it kind of took a while to discover Hop Latent Viroid, because you weren't seeing it throughout the plant. You might test a part of the hops plant, not find it, and then later on they did find it. So yeah, on your end, it's pretty similar. You're just throwing some leaf samples into paper envelopes and mailing it to the lab. And on our end, it's also pretty similar. We're going to take that leaf sample, the root sample. We're going to do a process called lysis, where we basically bust open the cells. So that can be done by boiling the cells, and then they burst open, and then that viral RNA will kind of float out into some solution. You can also physically smash the cells. What you'll do is like kind of freeze them with liquid nitrogen, get them really brittle, and then you bust them open with mechanical force.
Hmm.
So there's a couple different ways you can lyse the sample, and that whole process is just so you can get the virus and the RNA out of the cells, so that you can run a version of PCR to detect that RNA. So the PCR process is also different. For virus testing, viroid testing, I'm going to use that interchangeably. It's essentially the same thing. You do what's called a reverse transcriptase qPCR, or qPCR. And basically what you're doing is transcribing that RNA sequence into a DNA sequence. So the way the virus replicates is it gets read by your I don't know, maybe this is getting too into it. Your cellular kind of machinery basically reads that
Uh-huh.
RNA. RNA encodes a protein, or in this case, it encodes like another viroid, and it just gets replicated using your own cell machines. But anyway, yeah. So we're detecting RNA from the PCR sample instead of DNA,
Uh-huh.
and you just have slightly different reagents to do that. Uh, More reagents, different steps, but it's essentially the same thing as PCR, where if that RNA is there, it gets detected. We use qPCR, so that allows us to quantitatively test these. We can determine like relative levels of viroid between samples. We can say, okay, this sample has more, this sample has less, as opposed to just a positive or negative that you might get from from other PCR types.
Hmm.
Um,
All right.
and I think that's one thing that we're really proud of here at Farmer Freeman, is that we do have that, basically the standard, top of the line kind of technology. We're not gonna less chance of false negatives or false positives.
Right. Well, on that point, actually, we had a question that we wanted to ask about accuracy and reliability of these tests. Um, I'd love to hear actually for both the sex tests, because someone asked that in our community over the weekend or week when they found out that we were going to talk to you. So, the accuracy and reliability of both of these tests that you provide.
Sure, yeah. So I mean, validating the accuracy of the sex test is it's really simple. It's and obvious. You grow the plant and then you see what it grows. Does it grow pollen sacs or does it grow flowers? Yeah, for the most part, it's that simple. Sometimes though, the genotype, what we're testing, does not match the phenotype, which is what our customer grows and sees. And that can be kind of frustrating when you have a plant that's genetically male, but maybe it grows female flowers.
Interesting.
And so a lot of, I think every cannabis grower is familiar with like what they call them, Hermes.
Right.
Usually that's a female plant that'll grow male flowers, but it can go both ways.
I see.
Yeah. So our accuracy for the sex test, it's based on customer feedback over 6 years of testing, well over 99.5%. So
That's great.
less than 1 in 200 plants is you might see an issue with. And the
So
accuracy generally is inaccuracies and errors are coming from human error for the most part. It's not like the science and the technique is faulty. It's really just was the right sample put in the right tube, or did the
maybe
customer accidentally take 2 samples from the same plant thinking that they were moving from plant to plant, things like that.
or maybe
Even
you don't.
just inputting the information from one computer to another, you can have a typo. For the viroid, it's a little trickier because, especially with Hop Latent Viroid, because as the name implies, the latent name, you may not always see symptoms when the plant has it or not, or does have it. So you could get a negative test result. You could think that your plant is clean, and it could look clean, even though it is a positive infected plant. So that's a little trickier. But using the qPCR data, we have basically systems that allow us to verify, like, okay, our PCR worked. If it's not there, it's either because of a sample quality issue or that it's just not present in the plant. And so that's the importance of internal controls and using qPCR based systems. Other things for accuracy, I mean, for the viroid, it's really all about sample selection. If we're to test, one single blade of a top leaf, that may not be where the virus is on that plant. So it's important to get, multiple samples from around the plant. We're learning that, the upper root tissue is most likely to have the viroid, a Hop Latent Viroid. But for things like beet curly top, as that name implies, it's at the top of the plant. It causes curling of the leaves at the top of the plant. So yeah, just making sure that you're taking the right samples for the right test will help ensure accuracy. Other things, repeating testing is really important. If we ever see, like, maybe an unclear signal or any kind of irregularity, we'll repeat the test. We're not going to deliver results that we're not entirely sure that we did everything right, even if it means we're taking a loss on that specific order. Most important thing is just having, consistently accurate results and making sure that, you can remain a trusted laboratory. And then other things for reliability is just having standards and systems, following good laboratory procedures, making sure your equipment is maintained on a proper schedule, make sure your technicians are following procedure and taking notes and filling out their log forms, so that, anything can be verified should it need to be. Yeah, so sample, testing type, and just general business processes.
Great.
All right.
Well, let's
That's
get
good.
into let's kick off our section on HLVD. So it's been a hot topic. It's been a hot topic for sure. JR, did you want to officially kick us off? Because it looked like you had a comment there, but I stepped on you.
That's all right. We've got just a few viruses and viroids that we kind of want to go over.
Nice.
Just kind of like talk about what they are and kind of how you contract them and what possible symptoms might go along with that. And we might as well start with the dreaded HLVD. Can you give people an idea? I guess we talked a little bit about what it is. Can you talk about how it's usually contracted and what are its symptoms?
Yeah, sure. So I think HLVD is a great place to start because, I mean, that's what everyone's concerned about. And for for valid reasons. I mean, the symptoms from HLVD are arguably the most devastating from any of the other viruses and viroids that we can detect. You know, they can cause up to a 50% or more cannabinoid and terpene loss, in addition to looser and smaller buds. So not only do you have smaller, crappier looking buds, but there's also less cannabinoids and terpenes on those buds. It causes, you know, smaller leaves, malformed stems. People are noting brittle stems, making them harder to train the plant. And then some are saying clones won't root or grow as fast if they have HLVD. So yeah, Hop Latent Viroid, originally found in the hops plant, which is very closely related to cannabis. And now, you know, growers have dealt with dudded cannabis plants, you know, for decades. And now we generally attribute those dudded plants to HLVD. So yeah, that's a great place to start. It's definitely the most devastating of the pathogens.
Can you mention
Yeah.
actually I was talking about this with my wife and she said, "Well, when does it show up? You know, when does it start? When do the symptoms kind of show up? Is it when the plant is young? Is it when it's, you know, when I've been growing it for a couple months or whatever?"
Yeah, great question. It may not even show up. You can have positive infected plants that grow perfect. And that's what makes Hop Latent Viroid tricky, and why we need to keep learning more about it and to test more plants for it. There's you know, potential that the Hop Latent Viroid is a lot more widespread than we think it is, but we're just, you know, a lot of us are growing, you know, tolerant strains of cannabis that don't show those symptoms when they are infected.
Hmm.
Um, but generally with viruses, they'll kind of take advantage of when you are weak or when your plant is weak. So if you stress out your plant, you know, take a bunch of clones or cuts off of it, or when your clone is trying to root, basically when your plant is expending a lot of energy doing something, that viral will kind of take over, you know, see that gap uh, and take over.
So,
Yeah.
one last question I thought of before we get into, you know, so if I find it now, what do I do? But one last question that I can't remember if you mentioned earlier. Can HLV be passed through seeds? I've heard mixed things about it or whatever, and I've just wondered about that.
Yeah, I mean, I don't know for sure. I've heard some people say it can't, some people say it can. Um, yeah, I don't know the latest on that.
Okay,
You could definitely just, I would recommend everyone just proceed with caution when they are making seeds, test for everything, because cannabis cryptic virus definitely can pass via seed. Um, So if you're going to test your plants before you cross them, I think you should just test for hop latent as well,
that's a
and
good idea.
consider not breeding with that just in case.
Nice.
Okay, so now I've got my room. I've sent my tests off. They've come back hot. My bedroom has a, HLVD. What are going to be my protocols to eliminate that situation?
Yeah, I mean, the key word is eliminate. You probably want to get rid of those plants. Take them out back, shoot them.
Yeah.
Or if you are okay with the risk and your plants are looking good, keep growing them. I mean, it's really tough to say. I have customers ask me this all the time. I don't know exactly what is best for each individual. But I can tell you, you know, your plants won't always be duds just because they have the virus. Additionally, if we just got rid of every plant that ever tested positive, we're never going to be able to to identify and breed, resistant or tolerant strains of cannabis. Well, tolerant. I guess resistant wouldn't have the viroid at all, um, but that seems to be the only way out of this situation is to breed tolerant or resistant plants.
So is it kind of
You
like
can tissue culture.
Yeah, I was gonna ask about that. Yeah.
I was gonna say that is it like a virus thing like the flu virus where it's just gonna be there? It's gonna be something you're always gonna have to deal with and hope that you're doing your best to make sure that you're not getting sick.
Yeah, yeah, 100%. And yeah, it's not going to go away. It never went away in hops. What they did in hops is just they using breeding techniques found tolerant strains of hops, and that's how they deal with it.
Hmm.
Yeah, it's not going to be eradicated. It'll always be a thing in the industry.
So that's
So
where
my
getting these
recommendation,
protocols is important.
yeah,
Yeah.
my recommendations, I really do think in those cases, with the availability of good genetics around now, just kill everything
Yeah.
and completely sanitize and, do your best to make sure every bit of equipment, everything has been cleaned and sanitized and just start over. There's a lot of really good genetics out there with HLVD that you could bring back into your garden.
Well, and then also too, because I was talking about like I said with my wife, and I was explaining, why this is important or whatever. I was explaining, because there are farmers out there, or maybe even home growers, that they have strains that are really important to them, whether for their business or for their medicine. And so this is where it might be good if you have something that's really important. Don't just keep it around as a mother. Maybe make some seeds, or if you've got the cash, I guess, get a tissue culture done of it so that you can keep it around forever, and you're not going to be in the situation where you get this and then you're screwed basically.
Yeah, that's a great point. Yeah, if you keep mothers around, yeah, they can get sick. But if you self that mother and have seeds of it, yeah, keep the seed stash, and the seeds aren't going to get sick. Yeah, that's a great idea. Yeah, when it's a plant that's critical to your operation, then yeah, you're gonna want to probably tissue culture it out. The viroid's not going to be present in, I think, the meristem part of the plant. So basically, you just isolate that part of the plant and then regrow just from that part, and the tissue that grows will presumably be also virus free.
And
Um,
I've
yeah.
talked to folks that will do that multiple times, just to like maybe even 5 times over, just to kind of guarantee and make sure that that strand of HLVD is not present.
Extra clean. Wow.
Yeah, I don't know how necessary that is, but I've also not worked too closely with those people that do the tissue culture. I haven't been in their labs, running the hop tests right after just generally thinking that kind of sounds like
Yeah.
overkill.
So one last question on HLV before we get into our other viruses. We had a question from Big Bad Badger in the CannaBuzz community chat. Once again, shout out to the CannaBuzz community. Big Bad Badger said, "How does it get into your grow room? Is it airborne or what?"
Yeah, great question. It's not airborne as far as we know. It's always going to be transmitted mechanically, so that means touch. Uh, excuse me. Mainly using scissors, trim tools, things like that aren't properly sterilized. That'll transfer it from plant to plant. How you get it in general, like say you've never brought clones into your facility ever. I mean, it could be on someone's hands. They were working on a home grow. It could be maybe you have evidence that it was transmitted from seed. Um, but yeah, it transmits mechanically. No amount of air filters are gonna prevent it from going from room to room. It's all about not properly bleaching and sterilizing your tools and surfaces and
Yeah,
hands as you move from plant to plant.
interesting.
Yeah, we're gonna at the end of this, we kind of want to get into some of those best practices for cloning when you're taking off from your mothers and stuff like that. But now we want to move on to one of the ones that used to be the big deal right before HLVD, and that was the tobacco mosaic virus. Can you talk a little bit about how it's contracted and what its main symptoms are?
Yeah, so TMV, tobacco mosaic virus, that's going to be similar to HLVD, where it's transmitted from, you know, scissors, trim tools, plant to plant. Basically, the fluids and cuts on leaf surfaces transmitting from one to another plant. I think the most common symptom that you would see is the mosaic or spotting or patterns on the leaf surface itself. You'll see like areas that are yellow, areas that are light green, basically a mosaic pattern on the plant. As far as like how it affects yield and stuff, I'm not sure. You know, we just run the tests that people ask for. Uh we're not trying to like say like, oh, because we offer this, it's a really terrible virus.
Right.
But
No,
yeah,
it
so
makes sense that
yeah.
you're responding to demand, and I think that as you know, friends of you, you know, we've been in touch with you like we said for years. It's cool to see you adding all these different services and branching out, like we were saying earlier, into fig trees. That's really cool. So yeah, so people were coming to you and they were saying, "Hey, do you have this service?" Because it was impacting their crops, or at least they were worried about it, I guess.
Yeah, exactly. And yeah, so it's been challenging trying to, you know, keep up with demand for these different tests, but also understand why they want them. And then they ask follow-up questions. Well, what do I do? You know, that's three different people's jobs right there,
Yeah,
uh,
yeah.
at least. And you know, I'm the only scientist at our company, so forgive me if I don't have all of the answers. Uh, yeah, we're just
It's all good.
doing what people ask and doing it well.
No,
Yeah.
that's a big
Now
one.
you need to get your virus and viroid a consultant on your team and get out there and start consulting all these people who have hit your tests and had all these problems.
Yeah,
Um,
yeah,
so with
yeah.
the tobacco mosaic virus, just like any kind of the other ones, just maybe you see how it affects the crop and or maybe do the whole process of kill everything and
Yeah, it's one of those where if you detect it, I mean, there's not really much you can do. We don't have like antivirals that we don't have like pill that we can feed the plant. You know, you just eradicate the plant, and you know, maintain a clean operation and hope that it doesn't make its way back.
yeah. So the next one we're going to talk about is the cannabis cryptic virus, and I had really not really heard about that one that much. Can you maybe explain why people why that's on the radar and how you might contact it and what its symptoms are?
So from my understanding, CCV is not really related to any symptoms by itself. It usually is found like in tandem with other infections. So my guess is that it could exacerbate the effects of other pathogens such as you know, fusarium or botrytis. Um, you know, maybe if your plant has the CCV, it's going to be even more susceptible to bud
Hmm.
rot. Um, it's one of those viruses we offer the detection for it as like an add-on for our HLVD test. No one really requests CCV alone, but because
Okay.
it's a cheap enough add-on, people will go ahead and screen for it, and it's one that's known to be transmitted through seeds, so it's important for breeders to test for it. Um, but yeah, so we've tested for it a lot. We've actually never seen a plant positive for it. So we don't even have a customer that we can say, hey, how messed up did your plant look, when they
Right.
had this. So yeah, again, that one's a little more mysterious to us. It's not
Interesting.
as obvious as HLVD and why it's bad. Yeah.
Interesting. So, now let's talk about fungal infections. Um, do you offer services that test for that, or and also how do those manifest themselves in your plants?
Sure, yeah. So the most obvious fungal, I think pretty much everyone's experience is powdery mildew.
Hmm.
Um, and you just get that from having improper airflow and excess moisture, just all the general things on why you would ever have mold in any environment. Um, but the other ones that are also big deals are botrytis and fusarium. Botrytis being that gray bud rot, and fusarium being what we call root rot or wilt. And again, those are just all pretty much bad airflow, bad drainage. Fusarium can stick around in the soil, so my lab doesn't do it, but I'm pretty sure you can soil test for fusarium, and that may help you decide where to grow your plants. If you're looking into buying land, in addition to chemical tests for pesticide residue, you might want to do some biological tests for different pathogens such as fusarium. Um, being fungal, they have DNA, and we just detect for that DNA within the plant samples. Those tests are ones that we would also want multiple leaf samples for, just in case they're not present throughout the plant. Maybe it is at the beginning stage of an infection, where we might want to detect it in one part but not the other. Um, but yeah,
One
the
of the
thing, yeah,
Sorry, one of the things about powdery mildew that was always this a big discussion is it systemic? Can it get into the flow of the plant and get down into the roots? Is it only on the surface of the leaf, and does it get in
yeah.
cellularly? And
I don't know why that was ever really a discussion. I mean, the science and evidence I've seen wholly support that it's endophytic and that it gets inside of the plant and uses the vascular system. So yeah, that's why, even if it's not visible on the surface, your plant could still be infected with it. And why you would want to do a DNA test instead of just waiting around to see it take over.
yeah, and especially with taking in cuts. I mean, before we had places like King Kong clones or raw genetics clone sites where everything's batch tested, and you're getting something clean from a tissue cultured mom. Back in the day, those cuts that we brought in were more the culprit of our fungal infections than actually having airflow issues with filtering and stuff, in
Hmm.
my experience.
Uh, and Farmer Freeman, I had a question for you too before we jump into our next kind of topic or question. We had a question from Hunter Gatherer that came in towards the beginning of our stream that I thought was still relevant. So when I'm taking tests of leaves or whatever it may be with your tests, how like fresh do these need to be? If that makes sense, like, can I take a cutting of a cotyledon or the leaf or whatever, or like when you're doing with these tests, and put it in an envelope and then sit on it for a few days or a week or whatever? Or yeah, what's the deal?
Yeah. So with the way we provide our sample collection materials, they're all designed to preserve the samples. You don't need to like rush ship them. You don't need to worry about, temperature issues if you're in a cold or excessively hot environment. Um, I tell people, I have this HLVD positive leaf and an envelope that I've kept on my desk for almost 2 years now, and we'll periodically test it, and every time it still comes up positive. We'll dilute the sample 100 times and still be able to detect it. So the detection systems are really sensitive, and the plant leaf itself seems to not degrade, when it's in those envelopes, so everything kind of stays well and detectable.
Cool.
Because we don't have like CCV and tobacco mosaic known infected plants, we haven't validated that those can also sit on a desk for 2 years, but I would assume that they would still be detected. I can't imagine why they wouldn't be.
Alrighty. Well, thank you. So shout out to HG for that question. So yeah, let's get into talking about some best practices or suggestions that you have regarding cloning your mother plants. Like, what can I do to just make sure that I'm not going to run into any of these bad issues that we've just been talking about for the last, hour? So,
Yeah, a lot of people reach out to me. They're like, "Hey, I just took a bunch of clones, and now I want to test my mother." Well, you just did that in the totally wrong direction. Like, they're like, I don't want to pay to test all these clones." Well, you wouldn't have to, you test the mother first. Um, yeah, so definitely test your mothers before you work with them, before you take them into any kind of production rooms. Um, you know, Maybe work with just one mother at a time. If you're going to be working with multiples, just obviously use 10% bleach. So you get your jug of bleach, you take 1 cup of that, 9 cups of water. That's 10% bleach. So um, you're going to waste bleach if you just use it raw. It's not necessary. So, and it's cheap, so it goes a long way. So bleach will be your friend. It won't be friendly to your clothing or anything, but yeah, just get used to the smell of bleach. And so when you're using it, it's important to keep it on your tools and surfaces for about 60 seconds or more before you wipe it up. And generally just like a clean paper towel is sufficient to wipe it up. It will cause excessive rust on your metal materials, so be prepared for that. But yeah, I mean, taking cuts. If you are bringing new stuff into your grow facility, or even, I guess, planting seeds, because we don't know for sure if seeds can harbor it or not, keep anything new in like a kind of quarantined area. Don't be going between your quarantine and your production rooms, back and forth. Maybe working those on separate days. In quarantine for probably 30 days, and test, midway through that, or 3/4 of the way through that 30-day quarantine, so that the viroid has had time to reproduce, and be more likely to be detected.
wow.
Um,
That's
so yeah, I mean, you need to make sure your facility is designed around the quarantine, and not just have the quarantine as a side thought.
right. Yeah, really. I mean, my takeaway here is like, if you, especially for the the growers, the big time farmers, or if you've got you grow that you're paying the bills with, it makes sense to take this shit seriously and
Yeah.
to not fuck around, and to have these separate spaces and all these things, because it can really hurt you. It sounds like, and especially with everything that you've said, where a lot of these things, it can be hard to tell, and then all of a sudden you've got these dudding issues or whatever, and then you've got to start all over or whatever. You
Yeah,
can't pay the bills with a bunch of plants that are in compost, you know?
Yeah, I think once we have like tolerance markers and you know resistant strains of cannabis, you might be able to let your guard down a little bit if you know you're only growing, you know, strains that can handle the viroid. But yeah, because we don't know that information, yeah, you'll want to proceed with caution for sure. That makes me think when you do have an infected plant but it's looking good, definitely let us know because we want to DNA sequence that plant, see if there's any like weird variations that we know, okay, this this piece only exists in this plant that is resistant. Now we can use that piece, screen other plants to see if they have that piece, and you know start tagging different strains and lines as resistant or tolerant. Um,
That's
so that's
interesting.
critical. Yeah. Yeah.
How would you provide that information to the community? Just I would love to hear your philosophy because that's some you're touching on a little bit of like what Phylos used to talk about, but we kind of know what how Phylos ended up. But I'd be curious,
Yeah.
like if you start collecting this information, that's like
So
interesting identifiers like that.
yeah. So I mean, medicinal genomics is the lab that we work with for sequencing stuff. You'll have the option when submitting your samples, your DNA samples of do you want this publicly available or do you want to
Yeah.
keep your genetic sequence private? Um, so I imagine some of those resistant plants, people will say, yeah, let the genome be public. Uh, Others might not. Medicinal genomics, they're not gonna, if they find, you know, a marker that they associate with resistant plants, they're not gonna tell you that exact, like that's a secret sauce kind of detection marker.
Okay.
But when with that, you know, raw genome data being out there, you could find that marker yourself.
Yeah.
Um, I think that maybe
Yeah.
answers your question.
No, yeah, we could have a whole nother show on that whole topic. But
Yeah.
yeah, no, I appreciate that. Appreciate your
I,
thoughts on that.
I had one more thing about if I have multiples, like say I have 5 different mothers, it kind of makes sense that if I'm sampling each one, I would want to use different scissors for each one to make sure I'm not, doing any cross contamination.
Yeah, you can use bleach on your scissors between those plants. That should be fine. Swap your gloves between plants. Um, Another thing you can do is like razor blades are really inexpensive and they come in packs, and you just use a different razor for each plant. That can be a quick way of taking multiple samples. Um, the razor blades might not be branded as sterile, but they're not going to have like disease on the razor blades from the razor blade factory. So um, you should be good there. Um, One thing you could do is I would still recommend cleaning your tools before going from plant to plant. But if you wanted to save some money, we can combine like leaves from multiple plants into like one single detection reaction. And if it comes up positive, then we can test the plants individually to figure out where that positive came from. But that can be a way to screen multiple plants without paying for multiple tests. So that may be useful for if you suspect everything's negative, but just want to routinely test, that can be a good option for you.
So if I have 6 to test, I can maybe just send in 3 and probably figure my bases are pretty covered.
Yeah, yeah, like double up, you know, 2 plants per packet, and then if we detect something, then you could narrow it down from which actual plant has it.
Nice, nice, excellent. Well, let's do some final shout outs, shall we? Huh? I would like to have you tell us about any upcoming events, speaking events, any kind of shows that you're interested in and or will be at.
Yeah, sure. So our 420 sale ends tomorrow night. That's Monday the 24th at midnight. So I think any live listeners can still participate in this, but by the time this is published, I
Right.
think the sale might be
Yeah.
over.
Yeah.
But we do have that CannaBuzz coupon code.
Yeah.
So if it's your first time ordering, you can use the code CannaBuzz and get 10% off any of the testing services. Events, the one I'm looking forward to is CanMed. That's going to be down in Florida in May. And that's a really exciting one for the science community because it basically attracts like all of the big name cannabis researchers, cannabinoid researchers, doctors. There's going to be presentations about different HLVD variants and what that variation causes, what different symptoms those variants might produce. Lots of like high level talks like that. So we're really excited for that one. Otherwise, we're just kind of keeping an eye out. I think Virginia is going to have a big cannabis conference in October or in August. MJ BizCon, I'm considering maybe going to that for the first time ever this year. Maybe y'all should let me know if it's worth the hype. And then one thing I'm trying to do is go to more local grow shops and meet, growers directly in my area. So that's Virginia, Maryland, DC, even Delaware. I think they're talking about legalizing soon, so that might be a spot to go. But yeah, if anyone has events that they want to see me at, just reach out, let me know.
And I think that's a good reminder that if you got a local grow shop and you'd like to see Farmer Freeman easy sex testing in there, just hit him up and let him know to give Farmer Freeman a call. He'd like to get some retail kits out to any of your local grow shops.
Yeah, absolutely. We would love your help getting that. We're relying on grow shops to reach out to us at this time. So definitely tell them that you want it.
All
Great.
right. Well,
Well,
actually,
yeah.
I'll go ahead and take it, Q. I'll go ahead and take it. Where are all the places that people could find Jackson Farmer Freeman out there in cyberspace and beyond?
Yeah, sure. Website, farmerfreeman.com. Instagram, Farmer Freeman EZXY. On our website, you'll find my email address, our phone number. You can call, email me. We're just a 3-person company, so it's myself, my lab technician, and like an office manager who handles the shipments and a lot of like the basic customer service stuff. So when you call or email, like I'm reading it, I'm answering the phone. So don't be shy. Happy to talk and help and hear about your grow. And if you want to see me in person, invite me to some cannabis events. I'd love to show up.
Yeah.
Awesome. Well, Jackson, thank you so much to Farmer Freeman. Make sure to check out all their services. They're great. Like I said, I've used this at home myself. I'm just about to do some sex testing probably in the next week or so on some seedlings. You'll get a notification actually when the results show up. So you get a nice notification that like, hey, we got it. And then I've gotten an email then later that day already with results from some of the
Yeah.
samples. So I'll already like before that day know like half of the samples that I sent in what the results are. So it's kind of cool. Like there's some good instant gratification, as my ADD rattled, gamer brain. I like that instant gratification. And so there's that part of it too. But anyways, Farmer Freeman, thank you so much. Shout out to everyone in the CannaBuzz community. If you enjoyed this episode, please let us know by hitting the like button on YouTube. Leave us a comment below. Leave us something like growers love or support your local grower or support your local small business like Farmer Freeman. Let us know that you've enjoyed the show. Shout out to all of our CannaBuzz communities, our community members. Join us in our community. It's CannaBuzz. Search CannaBuzz in the App Store. It's a nice little community. It's like a Patreon plus social network, so you can post your grow in there, post your videos, or just hang out with JR and I. And then also shout outs to our friends over at Grove Bags. It's Terploc. these makers of Grove bags, great bags of all sizes. You can get, storage container sizes down to eighths. They make custom bags, all that good stuff. Grovebags.com. Use the code CannaBuzz for 7% off your order. And then, of course, TikiMadMan.com. Tiki Seeds. He's always got something new. And what was that latest cross he hit us with? It was called...
He's doing a whole big drop of the Gary Satan crosses, and he's got like 15 or more crosses in there, and those went live on 420. So check out those crosses there with the Gary Satan. And also next week we have Michigan Mac coming on. He's going to give us an update on the legal grow and the legal scene there in Michigan, and that'll be on April 30th from 2 to 3 p.m.
Nice. Well, live viewers, stay tuned. We're going to be giving away these pack of seeds, but for everyone else, we'll see you in the next episode. Thank you so much, Jackson, for hanging out on this Sunday. We hope you all have a great one. As always, support your local grower and
Growers love.